You and your genes. Animal watching at the zoo is a great way to spend an afternoon. There's a mother tiger and her brand new cubs. Wait a minute, one of the cubs has white instead of orange fur. How can that be? In every other way, the white tiger looks like tigers are supposed to look. It has black stripes, big paws with sharp claws, and a long tail. So what's the secret to the snow-colored fur? Its fur is white rather than orange because of a difference of a single gene. Genes are tiny structures inside cells that control the way cells grow and change. Genes are like a set of instructions for building living organisms and keeping them functioning properly. Whether it is a tiger or a toadstool, the cells of every living thing contain genes, and that includes you. Your genes are mostly responsible for how you look, from the color of your hair to the fact that you don't have a tail. It's true that your environment influences how you look and act, but genes play a big role. Genes are tiny, yet they are powerful. They must be if just one gene can make the difference between an orange tiger and a white one. In this book, you'll explore what we have learned about genes. You'll also learn what scientists are still trying to find out about these mysterious structures in our cells. Chapter 1. Life's Little Instruction Book. Tiger woods are attached. Tara's Lipinski's definitely are not. What are we talking about? Earlobes. Those soft little bits of flesh at the bottom of a person's ear. Notice how Tiger's earlobes are attached to the side of his head, but Tara's are detached? Her earlobes hang free. Which type of earlobe do you have, attached or detached? Grab a mirror and check it out. Like the color of a tiger's fur, the shape of your earlobes is caused by a difference in a single gene. Earlobe shape is a human characteristic or trait. We each have thousands of genes that control thousands of different traits. These instructions for some traits, like earlobe shape, are found in just one gene. Other traits are the result of many genes acting together. Has anyone ever said to you, you have your mother's smile or your father's nose? You have many traits that are very similar to your parents' traits because you inherited your genes from them. Half of your genes came from your mother and half came from your father. Those genes all came together in a cell that eventually developed into you. The set of genes that you inherited, your own personal set of instructions for life, are unique. Unless you have an identical twin, there is no one like you else on earth. There is no one else on earth exactly like you. In search of genes. Where are these genes that control so much of what makes you, you? They're inside almost every cell in your body. If you looked at one of your cells, say a skin cell from the tip of your finger under the microscope, you'd notice a dark blob near the center. That's the cell's nucleus. Inside the nucleus are long strands that are coiled up like tiny springs. These strands are your chromosomes. There are 46 chromosomes or 23 pairs in each cell body. One member of each pair came from your mother. The other member of each pair came from your father. Each of your chromosomes is made up of a substance called DNA, which stands for deoxyribonucleic acid. Scientists know that DNA looks a bit like a spiral staircase. Now, at last, we found the location of the genes. A gene is a particular length of DNA, a specific section of the spiral staircase that coils into the chromosome that lie in the nucleus deep within each cell. Interesting questions. Are genes found in every cell of a person's body? No. Genes are in every cell except blood cells. Red blood cells lose their nuclei and thus their chromosomes soon after they are formed. That's why red blood cells are shaped a bit like donuts. The squished in part in the center is where the nucleus once was. Question: does every human cell with a nucleus contain 23 pair of chromosomes? No. All cells with a nucleus except the reproductive cells, which are called eggs and sperm, have 23 pairs of chromosomes. Each egg and sperm cell contains just 23 individual chromosomes, not 23 pairs. When an egg and sperm join, the resulting cell can eventually develop into a person that has 23 pairs or 46 chromosomes. Working in pairs. Remember the examples of the tiger fur color and the earlobe shape? A single gene controls each of these traits. Actually, they're controlled by a single pair of genes. That's because people and tigers inherit pairs of chromosomes and therefore pairs of genes. From their parents, this means that for each gene on one member of the chromosome pair, there's a similar gene in the same place on the other member of the chromosome pair. How do these pairs of genes control a trait like earlobe shape? Well, the two genes for this trait that you inherited, one from each of your parents, may be different. Most genes come in slightly different forms called alleles. Different alleles are instructions for slightly different versions of the same trait. Two alleles are involved in earlobe shape. One is the dominant allele, and it's called for the detached earlobes. The other is the recessive allele, which calls for attached earlobes. The dominant allele gets its name from the fact that it can override or cover up the recessive allele. How does this work in you and other people? If you inherited two dominant alleles for your earlobe shape, one from each of your parents, you have detached earlobes. If you inherited one dominant allele and one recessive allele, you still have detached earlobes because the dominant allele overrode the recessive one. However, if you inherited two copies of the recessive form of the gene for each earlobe shape, your earlobes are attached. Complicating factors. If all traits were controlled by a single pair of genes, then genetics, the study of how traits are inherited, would be easy. However, it turns out to be very complicated. Many traits are controlled by more than a single pair of genes. The color of your eyes, for example, is the result of many pairs of genes working together in not a very clear-cut way. This fact makes it more difficult to figure out the job of each gene. It's true that genes play a major role in determining how an organism looks and functions. However, genes aren't the only influence. Environmental factors shape living things too. Take Siamese cats, for instance. Genes contain instructions for their color of fur, yet temperature also can affect their fur color. Where a Siamese cat's body is the warmest, the fur is the lightest in color. On slightly cooler parts of the cat, like its ears, paws, nose, and tail, the fur grows in darker. Now you think about your own body. Your genes are responsible for the fact that you have muscles in your arms and legs. However, you can make your muscles bigger and stronger by exercising. Things you do and experiences you have together with instructions given by your genes shape you into a unique person. Tracking down traits. These two pages show human traits that geneticists know are controlled by a single pair of genes. In each set of the photos below, the top photo shows the trait that results in a person if a person inherits one or two dominant alleles for the trait. Which form of these traits do you have? Tongue rolling, long eyelashes, widow's peak, hitchhiker's thumb. Solving a mystery. Sherlock Holmes, the famous English detective, pulled out his magnifying glass. He peered at the wall through the glass. Ah, a single fingerprint. It was that clue that would solve the case. The Sherlock Holmes stories are some of the most popular detective stories of all times. Holmes solved cases that stumped everyone else, yet scientists who study genes and how they work have been trying to solve a mystery that's far more complicated than any case Holmes ever took on. In the 1950s, scientists achieved major breakthroughs in the case of inherited traits. By then, geneticists had discovered DNA and figured out that it is made up of a chemical building blocks called nucleotides. Each nucleotide consists of sugar, something called a phosphate group, and one of four bases that were nicknamed A, G, T, and C. At this point, no one knows how DNA was actually put together. Researchers did know that DNA taken from every living thing had the same four bases. Maybe they had thought it was the way these bases were organized that was the key to the DNA's control over inherited traits. Maybe the bases formed a sort of code. Assembling the clues. The race was on to figure out the structure of DNA and break the genetic code. Rosalind Franklin, a young British scientist, provided a vital clue about DNA structure. She used X-rays to take some pictures of DNA. Those pictures gave American James Watson and Englishman Francis Crick the final piece of information they needed to figure out how the different parts of DNA fit together. In 1953, these two scientists showed that DNA is shaped like a spiral staircase with handrails on either side. The handrails are made up of sugar and phosphate groups. The bases A, T, C, and G form the stairs. Actually, each stair consists of two bases joining together, but not just any two bases. There's a pattern. Base A always pairs with T. Base C always pairs with G. Thousands and thousands of these base pairing form the stairs and a long spiraling strand of DNA. Passing it on. Once scientists knew the structure of DNA, they realized how it passes from one generation to the next. DNA can split down the middle and make copies of itself. Let's see how this works. When a cell is ready to divide, the long spirals of DNA unwind and the stairs, the base parts, come apart. Then two halves of the staircase separate like a zipper unzipping. Next, nucleotides are added to each old half of the staircase so that new stairs are formed. Wherever there's an A on the old half, the nucleotide with a T will pair up with it. Wherever there's a C, a G will pair with it, and so on. Stair by stair, a new half of staircase is built along each old one. When the last nucleotide slips into place, there are two spirals of DNA that are exactly the same as the original one. When the cell divides, two new cells form, and each of those cells contains a copy of the original DNA. When those cells divide, the same process repeats. With each division, DNA is copied and passed on and on and on. Cracking the code. What about the code? How is information about traits carried in DNA? The secret to the genetic code is in the bases A, T, C, and G. Long strings of these bases contain information used inside cells to make chemicals, mostly proteins. Proteins are the building blocks of cells. Your skin, muscle, and blood cells, for example, are largely made of protein. Your hair and fingernails, like a horse's mane and hooves, are made of protein too. It's the order of or sequence of bases along DNA that is the key to what kinds of proteins are manufactured by cells. For example, the sequence ATCCGAACTAG might code for one type of protein. A different sequence of bases, CGGGGTATAGCC, would code for a different protein, and so on. These different sequences of bases are separate genes. At last, the mystery of the genetic code was solved. Sequences of bases make up genes, and each gene contains the instructions for making a particular kind of protein. Inside a cell, those instructions are read, and proteins they call for are assembled. The newly manufactured proteins go into building and maintaining cells, and ultimately, the entire organism. Chapter 3. Imagine going to the library to look for a book about codes. To your surprise, the librarian tells you he's pretty sure there are books about codes somewhere in the library. However, he can't tell you where they are. There's no computer catalog to help you either. Yikes! What a mess it would be. Well, not that long ago, our understanding of human genes was pretty similar. True genetics had cracked the genetic code. Geneticists had cracked the genetic code. They identified a few genes, too, and located them on certain chromosomes. However, like a library full of books no one knew much about, our DNA remains largely a mystery. The idea of trying to identify all the genes in the DNA of a living thing is its entire genome seemed an impossible job. Then, in the 1970s, researchers discovered ways to read the sequences of bases in DNA more easily. Using high-powered computers, researchers began sequencing the genomes of different kinds of organisms. Scientists began with small organisms such as one-cell bacteria and microscopic worms. Even these tiny creatures had genomes made up of millions of base pairs. But by 1990, scientists were ready to try sequencing the human genome. They wanted to figure out the order of the 3 million or so pairs of bases that make up our DNA. The Human Genome Project. Scientists from many different countries joined in this incredible effort that came to be called the Human Genome Project, HGP. The genetic material for researchers used for sequencing was taken from several different people of different races. Using a mix was no problem because people are 99.9% the same genetically. In June 2000, after 10 years of hard work, researchers with some projects reached a huge milestone. They had sequenced enough human DNA to create a rough draft of the genome. Not every A, T, C, and G was figured out yet. However, enough base sequences were known for scientists to make some interesting observations. Some surprises. In early 2001, HGP scientists published some of their findings. One big surprise was discovering that humans have only about 30,000 to 35,000 genes. Scientists expected that we would have more since humans are large and complex. The exact number won't be known for sure until every base has been sequenced. Our genes come in different sizes. The average gene seems to be about 3,000 bases long. Some are much bigger. The largest gene sequence so far contains a whopping 2.4 million bases. We've apparently got some junk in our genome too. That's what HGP scientists are calling base sequences repeated over and over and over in the genome, which don't code for proteins. No one knows for sure what the junk DNA does. It may be involved in reshuffling the genome so that over time existing genes are changed and entirely new ones are created. Figuring out how many genes we have is a huge accomplishment, but many challenges remain. Scientists still don't know where our chromosomes, all our genes are located. Once we know that, however, we'll have a much better idea of where all the books are in our cellular library of life. Using new clues. How is knowing the location of every pair of bases in our DNA going to help us? How will figuring out each and every gene in its location on our chromosomes change our lives? One of the most important benefits of this research will be finding out the cause of many inherited diseases and finding ways to cure them. When genes are faulty and fail to do their job, cells and bodies don't work properly. The more scientists know about normal genes, the more easily they can spot defective genes and figure out a way to fix them. Fixing faulty genes as a way to treat diseases is a fast-growing area of genetics research known as gene therapy. Sequencing the human genome is also a big step towards understanding how DNA and proteins work with each other and with the environment to create complex living things. Researchers are discovering that base sequence in the DNA of many organisms soon will have a much clearer idea of how we are related to the other life forms with which we share our planet. However, understanding everything we find in that living library will be up to your generation to figure out. If you think you might want to be a DNA detective when you get older, don't worry, there will still be plenty of mysteries to solve.